Hello, everyone. Welcome back to Meds.Concepts. Today's lecture is on DNA repair mechanisms. Introduction to DNA damage and repair. DNA damage can occur due to both endogenous and exogenous factors. Exogenous sources include UV radiation, ionizing radiation, and chemicals. Endogenous sources include reactive oxygen species, replication errors, and spontaneous base deamination. These damages may alter DNA structure and interfere with replication and transcription. If unrepaired, they may result in mutations, chromosomal instability, aging, or cancer development. To prevent these consequences, cells utilize multiple repair systems. Major DNA repair pathways include direct repair, base excision repair or BER, nucleotide excision repair or NER, mismatch repair, translesion synthesis, and double-strand break repair. Direct DNA repair. The simplest DNA repair mechanism is direct repair. In this pathway, the damaged nucleotide is repaired directly without removing any base or nucleotide. One important example is photoreactivation. UV radiation induces formation of cyclobutane pyrimidine dimers, especially thymine dimers. These dimers distort DNA structure. The enzyme Photolyase binds to the thymine dimer and absorbs blue light energy. Using this energy, it breaks the covalent bond between adjacent thymines and restores normal DNA structure. Another example is alkyl group removal by MGMT, which stands for O6-methylguanine methyltransferase. MGMT directly removes methyl groups from guanine residues. Base excision repair, BER. Base excision repair or BER repairs small non-helix distorting lesions. Examples include uracil formation, oxidized bases, alkylated bases, and AP sites. BER is considered an error-free repair pathway. The pathway is initiated by DNA glycosylases. These enzymes recognize damaged bases and remove them by breaking the N-glycosidic bond. This creates an AP site, also known as an apurinic or a pyrimidinic site. Subsequently, AP Endonuclease cleaves the phosphodiester backbone near the AP site. DNA polymerase fills the gap and ligase seals the nick. Let us now examine the BER mechanism step by step. First, DNA glycosylase recognizes the damaged base and flips it out of the DNA helix. The damaged base is removed, generating an AP site. Second, AP endonuclease cleaves the DNA backbone near the AP site. Third, DNA polymerase inserts the correct nucleotide. Finally, DNA ligase seals the nick and restores DNA continuity. In eukaryotes, BER occurs through two pathways. In short patch BER, DNA polymerase beta replaces one or two nucleotides. In long patch BER, DNA polymerase delta or epsilon replaces several nucleotides, usually around 7 to 8 bases. Thus, BER efficiently repairs small base lesions while preserving sequence accuracy. Nucleotide excision repair. Nucleotide excision repair or NER repairs bulky DNA lesions that distort the double helix. Examples include thymine dimers, bulky chemical adducts, and UV-induced lesions. Unlike BER, NER removes an entire oligonucleotide segment containing the damage. NER is also considered an error-free repair pathway. In bacteria, 4 major proteins are involved: UvrA, UvrB, UvrC, and UvrD. NER recognizes distortion in DNA structure rather than recognizing a specific damaged base. NER mechanism in prokaryotes. In bacterial NER, the UvrA-UvrB complex scans DNA for distortions. Once damage is detected, UvrA dissociates. UvrB remains bound and locally melts the DNA around the lesion. Then UvrC joins the complex. UvrC acts as an endonuclease and creates two incisions. One on the 5' side and another on the 3' side of the lesion. Approximately 12 to 13 nucleotides are removed. UvrD helicase then removes the damaged oligonucleotide. DNA polymerase I fills the resulting gap and DNA ligase seals the strand. This restores the original DNA sequence. Nucleotide excision repair in eukaryotic NER in eukaryotes is much more complex and involves more than 25 proteins. There are two major forms: global genome repair or GGR and transcription-coupled repair or TCR. In GGR, lesions are recognized throughout the genome mainly by XPC protein. In TCR, stalled RNA polymerase triggers repair on actively transcribed genes. TFIIH plays a central role in DNA unwinding through its XPB and XPD helicase activities. Dual incisions are made by XPF-ERCC1 on the 5' side and XPG on the 3' side. Approximately 24 to 32 nucleotides are removed. DNA polymerase delta or epsilon fills the gap and ligase seals it. Defects in NER cause Xeroderma Pigmentosum, characterized by extreme UV sensitivity and increased skin cancer risk. Mismatch repair, MMR, in prokaryotes. Mismatch repair corrects replication errors that escape proofreading activity of DNA polymerase. Examples include mismatched bases and insertion-deletion loops. In E. coli, mismatch repair depends on DNA methylation. The parental strand is methylated at GAC sequences by DAM methylase, while the newly synthesized strand remains temporarily unmethylated. This creates hemimethylated DNA. MUT-S recognizes the mismatch. MUT-L acts as a mediator protein. MUT-H recognizes the nearest hemimethylated GAC site and creates a nick in the unmethylated daughter strand. Exonucleases remove the incorrect segment. DNA polymerase III resynthesizes DNA and ligase seals the nick. Eukaryotic mismatch repair. In eukaryotes, mismatch repair proteins are homologs of bacterial proteins. MSH proteins are homologs of MUT-S. MLH and PMS proteins are homologs of MUT-L. However, eukaryotes do not possess MUT-H and do not use hemimethylation for strand discrimination. Instead, nicks generated during lagging strand synthesis and interactions with pCNA help identify the newly synthesized strand. Mutations in mismatch repair genes are associated with hereditary non-polyposis colorectal cancer, also known as Lynch syndrome. Translesion DNA synthesis, TLS. When DNA lesions remain unrepaired before replication, replication forks may stall. To avoid fork collapse, cells utilize translesion DNA synthesis or TLS. TLS polymerase bypass DNA lesions and allow replication to continue. However, these polymerases have low fidelity and are highly error-prone. Therefore, TLS is considered a damage tolerance mechanism rather than a true repair mechanism. In E. coli, DNA polymerase IV and DNA polymerase V participate in TLS. Many TLS polymerases belong to the Y family of DNA polymerase. SOS response in E. coli. The SOS response in E. coli, which is a critical global response system that bacteria activate when their DNA undergoes severe damage. Step 1 and 2, Damage detection. The process begins when heavy DNA damage leads to the accumulation of single-stranded DNA, ssDNA, inside the cell. Recognizing this threat, a key protein called REC-A immediately binds to the ssDNA, forming active REC-A asterisk filaments. Step 3. LEX-A cleavage. Under normal, healthy conditions, a repressor protein named LEX-A stays bound to the DNA, keeping the SOS genes completely turned off. However, the newly formed REC-A asterisk filament triggers the autocleavage of LEX-A, causing the repressor to break apart and release its grip. Step 4 and 5. Activation and repair. With LEX-A cleared out of the way, the SOS genes are officially turned on and expressed. This pathway induces specialized, error-prone TLS polymerase, specifically polymerase 4 and polymerase 5, allowing the cell to bypass the damaged DNA and continue replication. Double-strand break repair. Double-strand breaks are among the most dangerous forms of DNA damage. They may be caused by ionizing radiation, replication collapse, or chemicals. Two major pathways repair double-strand breaks. The first is non-homologous end joining or NHEJ. NHEJ directly joins broken DNA ends without requiring homology. It is fast but error-prone and may introduce insertions or deletions. The second pathway is homologous recombination or HDR. HDR uses homologous DNA as a template and is therefore highly accurate. HDR mainly occurs during S and G2 phases when sister chromatids are available. NHEJ mechanism in eukaryotes. Let us now discuss the detailed mechanism of non-homologous end joining or NHEJ. NHEJ is the major double-strand break repair pathway active during G0 and G1 phases of the cell cycle. Unlike homologous recombination, NHEJ does not require a homologous DNA template. Therefore, it is faster but error-prone. The first step is recognition of broken DNA ends by the Cu70-Cu80 heterodimer. Cu-proteins bind to both DNA ends and protect them from degradation. Next, Cu recruits DNA-dependent protein kinase catalytic subunit, also known as DNA-PKCS. Together they form the DNA-PK complex. DNA-PKCS activates several downstream repair proteins through phosphorylation. If DNA ends are damaged or incompatible, Artemis nuclease trims the DNA ends to generate ligatable termini. Additional polymerase may also fill missing nucleotides if required. Finally, DNA ligase 4 together with XRCC4 seals the DNA ends and restores strand continuity. Although NHEJ efficiently repairs double-strand breaks, it often introduces small insertions or deletions, commonly called indels. Therefore, NHEJ is considered an error-prone repair pathway. Let us quickly revise some high-yield CSIR-NET concepts. Photolyase repairs thymine dimers. BER repairs small non-distorting lesions. NER repairs bulky helix-distorting lesions. UvrABC proteins participate in bacterial NER. XPF and XPG perform incisions in eukaryotic NER. MUTH recognizes hemimethylated DNA. TLS polymerase are error-prone. REC-A activates the SOS response. Cu70 and Cu80 participate in NHEJ. These points are extremely important for conceptual MCQs and PYQs. Thank you. If you found this video helpful, please like, subscribe and share with your friend.